Hello everyone. So today we're looking at B cell maturation as well as B cell activation as well as some functions. So here's kind of just the overview of what we'll be getting into. So here we can see we'll look at our B cell precursors, the function of our immature B cells, our mature B cells, and our activated B cells, which include our plasma cells and memory cells. And we'll also be taking a look at where these functions happen and how both negative selection and migration of B cells and antibody secretion works. So first off, B cells develop in the bone marrow, and then migrate to the secondary lymphoid tissues. So our mature naive B cells leaving the bone marrow travel to lymph nodes, the spleen, and Peyer's patches, which we discussed in our mucus lymphoid tissue lecture, which you can check out, and also our other lymphoid tissues in the malt. So again, we have our Peyer's patches in our intestines, our spleen, and lymph nodes, which are both secondary lymphoid organs. So we've seen similar figures to this before, but here we can see that the early stages of B cell development rely on the bone marrow stromal cells. So here we have the different phases. We have our multipotent progenitor cell. Then we have our common lymphoid progenitor, early pro-B, late pro-B, pre-B, and our immature B cell. Multipotent progenitor cells express FLT3. So FLT3 binds to FLT3 ligand, or FLTL3L, on the stromal cells. So here's this is our bone marrow stromal cell. So we can see FT3 binding to FLT3 ligand. This causes signaling through the FLT3, and this is required for the differentiation to the next stage, which is the common lymphoid progenitor, as we can see here. So then our stromal cells make CXCL12. So this chemokine helps keep progenitor cells associated with the stromal cells. So again, we have our different types of chemokines, and this is one of the nomenclature. And we also have IL-7, which binds the IL-7 receptor, which creates signaling, which promotes survival. So pro-survival signal, here we can see our IL-7. These progenitor cells bind to vascular cellular adhesion molecule 1, or VCAM, on the stromal cells, and this promotes the binding of KIT, or also known as CD117, on the pro-B cell surface. And this allows for pro-B cell to bind to the stem cell factor, or SCF. Here we see our KIT and stem cell factor binding. Stem cell factor, SCF, binds to KIT. This results in kinase activation and induces B cell progenitor proliferation. So these pro-B cells mature into our pre-B cells and then into our immature B cell. So here's a similar picture going through the stage, but it's looking at our gene rearrangements that occur at the different stages. So here we have our heavy chain genes, light chain genes, and surface immunoglobulin. B cell development has several stages marked by immunoglobulin gene rearrangement and expression. So our heavy chain gene rearrangements take place in pro-B cells. We can see here's our pro-B cells, and are tested using the pre-B cell receptor. So successful pre-B cells are stimulated to proliferate, becoming large pre-B cells, and these progress to resting small pre-B cells. And we have pre-B cell receptor expression. It stops and is rearranged. We have our rearranged light chains using the RAG proteins, as we've discussed, and VDJ recombinase. This successful rearrangement results in immature B cells, which express IgM at the cell surface, and mature B cells produce IgD and IgM by alternative mRNA splicing. So again, as we said, our naive mature B cell has IgD and IgM, while the immature has IgM only. And again, there's ongoing changes in gene expression at different stages of B cell maturation. So here's all the different proteins, and essentially the darker the color at each stage, we can see where it's happening. So again, here are a bunch of different proteins that are involved in B cell differentiation or gene expression. So expression of cell surface receptors, which include many of these listed, enzymes and transcription factors at each stage of B cell maturation allow for certain activities to be carried out at defined stages. So we've seen this before, both RAG1 and RAG2. We've seen our TDT, and here we can see their function. So RAG1, RAG2, lymphoid-specific recombinase, TDT is an N-nucleotide addition. Here we'll talk about these later, but there's lambda-5 and V-pre-B, which involve in surrogate light chain components. Here we have our signal transduction factors. Here we have our growth factor receptors. Remember, we talked about IL-7, helping with survival. And we also have BP1, which is aminopeptidase. B-cell stages can be identified based on expression of distinct receptors. And transcription factor, as we can see here, an example would be the pre-B cells, which no longer express KIT and IL-7 receptor. So we can see here, here's our IL-7. After you reach here, no longer IL-7. They now know to survive, essentially, after that point. So just kind of an overview of the B-cell rearrangement or immunoglobulin rearrangement we've seen. So you can go back to our B-cell lecture if you want more detail. But these genes are productively rearranged immunoglobulin genes, which are immediately expressed as protein by maturing B cells. So in the early pro-B cells, the heavy chain gene rearrangement is not yet complete, and there's no functional mute protein is expressed. Only transcription occurs. So this productive H-chain gene rearrangement results in mute chain expression in complex with the surrogate light chain components. These were their V-pre-B and lambda-5, as we talked about on the last slide. And these form the pre-B cell receptor. Again, we have immunoglobulin A, alpha, and beta, which are also expressed with the B cell, as you can see here. And this allows to transduce signals to the B cell to stop the heavy chain rearrangement and proliferate. And then we have a transition to our pre-B cell stage, where the light chain gene rearrangements take place. Successful L chain production results in is due to the combination with heavy chain gene, and this results in IgM on the cell surface, again with Ig alpha and Ig beta, to form the B cell receptor complex. So this signaling stops the light chain gene arrangement. So here in our early pro-B cell, we see VH to DJH rearrangements occur in the late pro-B cells. Now we have our large pre-B cell. Then we have, this stops the heavy chain gene rearrangement and progression to light chain gene rearrangement in the small pre-B cells. Then we have our immature B cell. So again, you can see the differences here between these two. Then we have the stop the light chain gene rearrangement in our mature B cell. So immunoglobulin gene rearrangements at different stages of B cell maturation are controlled by stage-specific expression of different enzymes. So expression of the B cell receptor components is also stage-specific. So our surrogate light chain components are V-pre-B and lambda-5, which you can see here, surrogate light chain components. And they're expressed as the B cell transitions from pro-B cell to pre-B cell. So again, here's our pro-B cell to pre-B cell. We kind of see the stages here. So immunoglobulin alpha and Ig beta are needed for successful signal transduction through the pre-B cell receptor and B cell receptor. This pre-B cell receptor dimerization causes a signal which helps progress to light chain gene rearrangements by the pre-B cells. So like in the last slide, our heavy chain rearrangement stops, then we switch to our light chain. So we have a kinase called BTK, or Bruton's tyrosine kinase. We'll see this again later, but this is needed for successful signaling. So BTK mutations can prevent B cell transition from the pre-B cell stage and cause lack of maturation of B cells. So this is seen in some diseases like X-linked agammaglobulinemia, which is a BTK mutation, also known as XLA. So stages of maturation and differentiation. So we've gone through some of these before, but we'll take a closer look. So at the stem cell stage, there's interactions with bone marrow, stromal cells, like we showed before, cytokines, and growth factors, and these provide signals needed for survival and maturation in the next stage. So the next stage is our pro-B cell. The pro-B cell is the earliest stage in B cell lineage, so that's important to remember. And there's immunoglobulin gene rearrangement begins. So first we have our heavy gene rearrangement, which takes place. Then we have our pre-B cell, where the pre-B cell initially expresses a pre-B cell receptor on the surface to test the mu chain. So this mu chain and surrogate light chain and Ig alpha and Ig beta form the pre-B cell receptor. If this is successful, there will be proliferation and then light chain rearrangement. Then there'll be light chain rearrangements which occur. This pre-B cell receptor signaling causes allelic exclusion and stops further heavy chain rearrangement. Again, we talked about BTK, or Bruton's tyrosine kinase, which is involved in cell signaling through the pre-B cell receptor. The pre-B cell receptor has a mu-H chain associated with a surrogate light chain made up of V pre-B and lambda 5, and is associated with signaling components Ig alpha and Ig beta. So here's our V pre-B and lambda 5, and then our Ig alpha, Ig beta. So from the stem cell, we have interactions with the bone marrow, stromal cells, cytokines, growth factors, which provide signals needed for survival and maturation in the next stage. As we said, then when there's a pro, then there's a pre, and again, if it's successful, we have this light chain rearrangement. So after this, we have our immature B cell, where the light and heavy chains pair. This forms the monomeric IgM, and this is expressed on the surface, and then these B cells undergo selection processes to delete self-reactive B cells. Then we have our mature naive B cell, which expresses both IgM and IgD on the surface. After immune antigen stimulation, this naive B cell will be activated. Then we'll have our memory B cells, or plasma cells. So these plasma cells are the ones producing antibody. So what is the pre-B cell receptor? So here we can see the pre-B cell receptor. So things we talked about are V pre-B and lambda 5, which again are a surrogate light chain, and Ig alpha, Ig beta. So the pre-B cell receptor uses invariant V pre-B and lambda 5 as substitutes for the light chain to test the ability of the mu heavy chain to successfully interact with the L chain and form a B cell receptor. The V pre-B substitutes for the light chain variable region, and the lambda 5 substitutes for the light chain constant region. So V pre-B for the V, so that's kind of easy. V for L, sorry. And then, yeah, so the light chain variable region, so these are all light chain. So again, light chain surrogates. And then lambda for C, which is not as easy to remember. So yeah, V to V, lambda to C. Oops. So the pre-B cell receptor resembles the B cell receptor, but the difference is that the pre-B cell receptor does not have a true immunoglobulin light chain. Instead, it uses these surrogate light chains made up of V pre-B and lambda 5 proteins. So what does this pre-B cell receptor do? So this pre-B receptor uses invariant proteins, like we said, which act as substitutes to test the ability of the mu heavy chain. So successful pre-B cell receptors form dimers, results in dimerization and signal transduction. This is through associated chains of Ig alpha and Ig beta, which have ITAMs, which we've talked about quite extensively. So here's our ITAMs on Ig alpha and Ig beta. And this involves BTK, which is Bruton's tyrosine kinase. This results, if there's successful signaling, there's progression through the pre-B cell stage, then there's proliferation, followed by light chain gene rearrangements. If there's a lack of success, we have apoptosis, where the cell is removed. So BTK mutations, like we said, can prevent progression through B cell from the pre-B cell stage to immature B cell stage. So again, we won't have any B cells. So B cells. The B cell rearranges its heavy chain locus first, like we said, and our pro B cell has a D gene and a J gene segment, which are randomly joined. So we have our D to J joining, forming our DJ segment. Next, we have a variable heavy, or VH gene, is joined by the DJ segment, forming the VDJ joining and the VDJ segment. If this is productive, again, if, this produces a protein that can be transcribed and translated into a mu heavy chain. This cell progresses to a pre-B cell stage, and the heavy chain gene rearrangements are ended. Then we test this. This is tested using the pre-B cell receptor on the surface. So there's mu chain and surrogate light chain and Ig alpha forming that pre-B cell receptor. So if it was not productive, this pro-B cell tries heavy chain gene rearrangements on the second chromosome. And if it works, testing is used for the pre-B cell receptor, and this progresses to the pre-B cell. So if, again, we go from our kappa to our lambda, so that's kind of what's happening here. So if it doesn't work, we'll move to the lambda locus on the other chromosome, and then if not, then we'll have apoptosis. B cell dies by apoptosis. So the steps in immunoglobulin gene rearrangement were where developing B cells can be lost. So heavy chain gene rearrangements are carried out first, then our kappa L chain, like we said, and then the lambda L chain. So kappa, 1 kappa, 2 lambda. That's bad lambda symbol. So more cells are lost at the pro-B cell to pre-B cell transition stage than at the pre-B to immature B cell transition stage. This is because the B cell has more opportunities for successful rearrangement when leaving the pre-B cell stage. So again, this is just a figure if you want to follow it. So here are the rearrangements down here at each stage. So again, early pro-B, H-chain rearrangement. Still have H-chain rearrangement. Then in the pre-B, we have our light chain, and then a rearrangement ceases. So it's past the test at this point. So again, if we can see here, these are our positive feedback. So if we do have rearrangement here, if it's successful, so yep, we're successful. Then we move here. Do we have a kappa gene that works? Yes, goes here. If it doesn't work, we try again, rearrange kappa gene on a second chromosome. Does it work here? No. If it's yes, then we go here. And again, if nope, then we try our lambda gene twice. And then if not, cell loss. So essentially it has multiple tries at being successfully rearranged. 2 for the kappa, 1 for each chromosome, and 2 for lambda, 1 on each chromosome. And if none of those work, and they're either self-reactive or they don't bind strongly, they're removed through apoptosis. So B cells with successful heavy chain rearrangements. The next try light chain rearrangements. So again, they try kappa light first with the VJ segments joined. If this is successful, the immature B cell with IgM on the surface will be a kappa light chain. But again, the cell has 2 chances for each one, so it can rearrange kappa on the second parental chromosome if the first is unsuccessful. So this is through ileic exclusion, which prevents rearrangements on the second chromosome if the first try was successful. So this is important. If the kappa L chain formation was not successful, it will try the lambda light chain. Then again, if that's successful, we'll have our immature B cell with a lambda light chain. And if it's not successful, it will try again one more time on the other chromosome. Then if not, it dies by apoptosis. Cells that failed at either checkpoint die by apoptosis, like we said. So essentially, this is kind of our 2 checkpoints. So we can see here, no pre-B cell receptor, apoptosis. No B cell receptor, apoptosis. So here we have commits to B cell lineage. Then we can generate heavy chain diversity in the pro-B cell population. And then here, this selects for functional heavy chain, so no functional heavy chain. If it doesn't work, apoptosis. Then we generate the light chain gene diversity in the pre-B cell population. Then again, we select for our functional light chain. Don't have a proper functional light chain, remove through apoptosis. And then if they all work, we have a functional IgM and a proper functioning B cell. As we said, 2 key checkpoints in the B cell maturation in the bone marrow that detect whether a functional B cell receptor is made. The first checkpoint tests for a functional heavy chain, and the second tests for a functional light chain. This is a good figure if you go over again if you want to take another look. So, removal of autoreactive B cells. There are several different possible fates for B cells undergoing maturation. So if there's no self-reaction, the B cell matures and migrates out of the bone marrow, as expected. If there's a strong self-reaction, there'll be clonal deletion by apoptosis and rescue by receptor editing. So we can detect that it is self-reactive, and then we can actually rearrange it again so it isn't. We can also have weak self-reaction or soluble antigen, which lacks soluble IgM and cross-linking. So these B cells migrate out but are anergic and have a short lifespan. So essentially, they really can't bind to anything due to the lack of soluble IgM and cross-linking. And then they become anenergized, so they're not functional. And then eventually they die due to lymphocytes having relatively short lifespans. But B cells also undergo testing in the spleen. And low affinity self-reaction can lead to immunological ignorance. So no reaction. So antigen becomes inaccessible, and there'll be lower weak affinity. So again, there's no self-reaction, migrates to periphery, mature B cell. We have a multivalent self molecule, So this is our self molecule here. There'll be clonal deletion or receptor editing, and there'll be apoptosis or generation of a non-autoreactive B cell. We have a soluble self molecule. This migrates to the periphery and is anergic. We have low affinity non-cross-linking self molecule. Again, migrates periphery and is a mature B cell that is clonally ignorant. So several different possible fates for B cell undergoing maturation. So we have our energy. So when there's weak self-reaction, as we said, the B cell can migrate out but are anergic. These B cells have little or no surface IgM and do have surface IgD, but does not activate the B cell. So these enter the circulation, and through the short lifespan of 1 to 5 days, they die. So this IgM of immature B cell binds soluble univalent self-antigen. The B cell signal to make IgD and to become unresponsive to antigen, and then enters the peripheral circulation but does not survive for long and ends up dying. But again, it's anergic. So responses of an immature B cell to self-antigens in the bone marrow. So if there's an interaction with a cell-bound antigen that reactivates the VDJ recombinase using the RAG proteins or receptor editing. So this generation of a receptor reactive self-antigen results in deletion of the B cell. Generation of a receptor reactive to non-self-antigen allows the B cell to leave the bone marrow. So this is our rearrangement to make it non-reactive. If there's interaction of the immature B cell with the soluble self-antigen, this results in anergy and long-term inactivation. So here's kind of the process. Here's our self-antigen. Again, if we have receptor editing, reactivation of VDJ recombination at the L-chain locus. For the L-chain recombination, now we have a B cell expressing non-self-antigen, and this B cell will leave the bone marrow. This detects so if we, again, we can either have receptor editing or anergy. So if it decides to be energized, there'll be long-term inactivation, and the activated B cell will leave. So we can also have B cell expressing self-antigen. So even though we had recombination here in this step, it's still self-reactive. So again, it's still looking for self-antigen, apoptosis, and deletion. Immature B cells undergo a selection process in the bone marrow to remove self-reactive B cells. Successful B cells that are not self-reactive can become mature B cells. So receptor editing. So replacement of the L chains by receptor editing can rescue some self-reactive B cells by changing their antigen specificity. If a strong self-reactive signal is received through the B cell receptor, cross-linking of the soluble IgM results in B cells do not turn off RAG gene expression, and then these RAG proteins continue to facilitate light chain gene rearrangements, and there'll be a new L chain is expressed, and this combines with a heavy chain to make a new B cell receptor. So here we see our strong ligation of IgM by self-antigen. There'll be arrest of B cell development and continued light gene rearrangement with low cell surface IgM, and then a new receptor specificity is now expressed. You can see it's represented here in a different type of receptor. So if this new B cell receptor is not autoreactive, B cell is therefore rescued, and the B cell matures and leaves the bone marrow. But if this new B cell receptor is autoreactive still, either to a new antigen or the same, B cell continues to cycle the L chain gene rearrangement until either it is rescued or is deleted by apoptosis. So here's our new receptor being expressed. So again, it can be new. If it's still self-reactive, the B cell will die. But if it's no longer self-reactive, the immature B cell migrates to the periphery and matures. So just summary, this is just the last slides into one if you want a summary. So I'll just leave it. I won't write on it too much if you want to pause and take a look as a recap. So I'll just write here as a reminder if you're going through it, recap. So B cells that bind to self-antigen are removed from the repertoire of B cells with randomly generated binding specificities, while non-reactive B cells are permitted to enter general circulation. Again, here's this nice summary figure if you want to go through it. So B cell signaling. B cells become activated when the receptors are cross-linked by antigens. So again, cross-linking is very important. So here you see a bunch of... So here we have an antigen-presenting cell, which is activated by a T helper cell, activating a T helper cell, which in turn activates the B cell through signal 2. Here's signal 2. And the B cell differentiates into antibody-producing plasma cells. So cross-linking of antigen receptors is the first step in B cell activation. That's very important. First step. So again, cross-linking. First step, B cell activation. Just went over this again just so you guys can remember. So, the B cell receptor is part of the immunoglobulin superfamily. The B receptor does not work alone. So, in the B cell structure, we have our 2 heavy chains and our 2 light chains, and an antibody molecule with the transmembrane region. So, here's our B cell receptor. So, we need our Ig alpha and Ig beta as well, which has the signal transduction molecules, which we discussed are ITAMs, which are immunoreceptor tyrosine-based activation motifs, which are used for signaling activity. We also have ITIMs, which we'll discuss, which are immunoreceptor tyrosine based on inhibition motif, which downregulate signaling, which we saw in T cells and other pathways. So the membrane form of antibody. So membrane immunoglobulin, which can also be seen as just like MIG, membrane immunoglobulin, serves as a B cell receptor for antigen recognition. It has a signaling role. So the cytoplasmic tails of the Ig alpha and Ig beta complexes recruit kinases, as we said, ITAMs, and this results in intracellular signal transduction after antigen binds to the membrane immunoglobulin. So again, here's our Ig alpha and Ig beta with our kinases. So B cell receptor attachment to antigen results in cross-linking. So here's 2 antigens cross-linking on an two receptors, cross-linking on an antigen. This results in activation of a family of kinases called FIN, BLK, and LIN, and these phosphorylate the ITAMs we discussed on Ig alpha and Ig beta. There's also SYK, which is a kinase which binds to the phosphorylated ITAMs of Ig beta. So here, phosphorylation of the ITAMs on the B cell receptor tails by SYK family kinases, which are these kinases right here, FIN, BLK, and LIN. And then we see here SYK binds to the doubly phosphorylated ITAMs and is activated on binding. Signal transduction via the B cell receptor. So when the B cell receptor is cross-linked, which again is the first step, kinases associated with the B cell receptor complex phosphorylate tyrosines in the ITAMs on the cytoplasmic tails of Ig alpha and Ig beta. Other kinases can now interact, and we'll go through this figure slowly. So again, this is kind of where we're going. We'll have the other one in the next slide. So B cell receptor attachment to antigen causes, again, our cross-linking and then kinase activity. Again, note the cluster of receptor complexes, so we can see here. So this activation of kinases, including FIN, BLK, and LIN. Again, phosphorylated ITAMs on Ig alpha, Ig beta. Then SYK binds to the phosphorylated ITAMs of Ig beta. Then SYK transphosphorylates more SYK and other targets. So signal transduction. So again, this is essentially what we said, but here we can see here. So once SYK binds to the phosphorylated Ig beta ITAMs, which we can see here, and since there are at least 2 receptor complexes per cluster, so here's 2 in this example, 2, SYK can phosphorylate other SYK and cause further signaling. So this SYK is phosphorylating the other SYK and vice versa. So next, additional kinases and adapter proteins are recruited, and these amplify signal transduction processes, which allows more adapter proteins to be recruited. This results in the activation of intracellular signaling pathways. So again, here's the whole process right here. So again, I recommend going through this slowly if you want. And then this will involve phospholipase C activation, which we've seen before, PLC gamma. You can see here. And then this results in diacylglycerol, a DIG, and IP3. So we've seen this pathway before with the other activation, where it ends up splitting. So again, here we have our DIG, and then our IP3, and then we've seen this before with calcineurin. Then here we have our protein kinase C, so it's similar to the one thing we saw before. We also have MAPKs. So again, we have our diacylglycerol resulting in protein kinase C activation with NF-kappa B activation and cytokine transcription. And again, remember that all of this stuff we saw with T cell receptors was also related to the transcription of all these transcription factors, NF-kB, NF-kappa B, NFAT, and AP1. Then we also have our IP3 pathway, which involves in the mobilization of intracellular calcium, as we discussed, and the activation of calcineurin, which is a phosphatase and results in NFAT activation. Then MAPK, or MAP kinase, has cascade activation and activates AP1. Again, the overall outcome is cell activation and the activation of these transcription factors and gene expression and B-cell proliferation. So yeah, definitely recommend this figure. It's kind of a lot of overlap with the other one we talked about, but a lot of this stage up to here is different. So here's kind of our summary. So first, we have our B-cell cross-linking, which again is the first step. And then we have our kinase activity, then our kinase activation. Then we'll have signaling complex assembly and the activation of intracellular pathways. Then we have the activation of our 3 transcription factors, which again were NF-kappa beta, kappa B, and NFAT, and AP1. You see that here? This is another good figure if you want to take a look at it. So we have our cross-linking, the activation of our kinases, Lin, F, BLK, and FIN. This activates SYK, and this SYK causes our adaptive proteins. Then we have the formation of our PLC, phospholipase C gamma, which activates DAG and our IP3. And then here's our DAG pathway, our IP3 pathway with calcineurin and elevated calcium, and then our MAP cascade, resulting in the transcription factors we've seen. And then in the end, we have cytokine receptor and immunoglobulin genes being produced. And then as we see, immunoglobulin and cytokine receptor gene transcription. So again, these gene expression are for the encoding of cytokines and receptors and antibodies. So our B-cell receptor complex. So after all that, we still have other signals that need to be talked about. So that's just for our B-cell receptors. So now we have to look at our B-cell co-receptors. So again, our B-cell receptor complex was our B-cell receptor and the Ig alpha, Ig beta. But now we have our co-receptor, which is a complex of 3 proteins, CD19, CD21, which is also known as CR2, which is complement receptor 2, and CD81, which is the squiggly thing there. So the roles of this co-receptor are to amplify the B-cell receptor signaling. So similar to T-cell receptors where we had B7 to C28, CD28, this is kind of a similar function but different molecules. So this was T-cell. This enhances signaling efficiency of signaling the process. So we need less antigen, which is needed to activate the B-cell. So although we need a cross-linking, we'll need less. So this works because antigens are often coded with complement proteins, including C3D. And as we said, we have a complement receptor, CR2, which is a receptor to C3D. So we see this bacteria with a bunch of C3D fragments. So then we basically need less antibody to bind to get all their activation. But we need our co-receptor as well as this B-cell receptor interaction. So the B-cell co-receptor, so we have CD19, which activates intracellular signaling when CD21 or CR2 is engaged by complement fragment C3D. C3D is a product of C3b and has been called a molecular adjuvant due to its ability to make antigens more immunogenic. So essentially it makes this antigen stimulate our immune response more strongly. So now we're going to be bringing back complement like we just discussed. Guys might not want that, but we need it. So antigens are often coded with complement proteins, like we said, C3D. And we have CD21 and CR2, which is the receptor for C3D. We have complement receptor 1, which is another complement receptor on B cells, facilitates C3D production. So here's our CR1, we'll talk about, and then there's CR2. C3D is a product of C3b, generated when CR1 it binds to C3b, and then it's cleaved into C3D. C3D is coded antigen A C3D-coded antigen can cross-link with B-cell receptors with co-receptor complex, causing phosphorylation of CD19 by the B-cell receptor-associated tyrosine kinases. So here we see our complement receptors, IC3B and C3D, IC3B and C3D, which bind to the CR2 CD21. We can see that here. This allows the co-receptor to sense pathogens, essentially. And as we said IC3B and the C3D are products of C3B. So this process is facilitated by another complement receptor, CR1. Here's our CR1 receptor again, which CR1 binds to C3B fragments on pathogen surfaces, which again, we've seen this as an opsonin, so we've seen this many times in our complement pathway, making them susceptible to cleavage of complement factor 1, which is a serine protease, generating IC3B. IC3B is cleaved to the produced C3D. Both C3D and IC3B bind to CD21, or CR2. Yep, that was a mouthful. But yeah, guys, you can go back and slow it down if you need. But yeah, it gets a little confusing. But again, I think the best thing you could do, read the text or go through the figure first and just follow the headings in the text and look at the figures. So, complement fragment C3D coded antigens can cross-link B-cell receptor with co-receptor complex. This results in the phosphorylation of CD19 by the B-cell receptor tyrosine kinases. This leads to the Circ family kinases, which augments signals sent through the B-cell receptor. So again, the B-cell receptor and co-receptor cooperate in B-cell activation. These signals generated from the B-cell receptor and its co-receptor synergize, so again, that's important, to activate B-cells. CR1 or CD21, binding to C3D fragments on the pathogen surface, clusters the B-cell's co-receptor complex with the B-cell receptor. The cytoplasmic tail of CD19, as we can see here, it is then phosphorylated by tyrosine kinases. So here's our tyrosine kinases associated with the B-cell receptor. Phosphorylated CD19 then binds to intracellular signaling molecules, and these signals synergize with those generated by the B-cell receptor, causing signal amplification. So again, this is essentially the whole purpose of the whole process. Activation signals. So this is essentially a similar picture, but this is in a little more detail. So now we can see what happens further down. So a short recap. Our complex of antigen and CD3 binds to the B-cell. Antigen binds to the B-cell receptor. CD3 binds to C3D binds to CD21 or CR2. The result is clustering of the B-cell receptor and CD21, resulting in the phosphorylation of CD19 by B-cell receptor-associated tyrosine kinases. This leads to the recruitment and activation of additional kinases, which augments signals that send through the B-cell. The role of this is to amplify B-cell receptor signaling to enhance efficiency of signaling process, meaning we need less antigen to activate the B-cell. So that's kind of the main purpose. Amplification, so we need less. Signaling through the B-cell receptor, co-receptor lowers the threshold for antigen needed to activate B-cells through the B-cell receptor. So here we can see a more detailed pathway. So here we have our binding of CD21 to the C3D tag antigen, which allows for co-receptor to cluster with the antigen receptor. Co-ligation of the co-receptor allows receptor-associated kinases to phosphorylate CD19. Phosphorylated CD19 binds to the Circ family tyrosine kinases, for example, Lin and PI3K kinase. PI3K initiates downstream signaling pathways. So this is a really good figure here to essentially sum up what we would see on a B-cell surface. So there can be multiple of these on the same cell, but it kind of gives you definition of each one. So here's our IG, heavy chain, light chain. So again, here's our B-cell receptor components. We have our CD21, CD19, and CD81. So these are our co-receptors, which result in additional signal transduction molecules associated with the B-cell. We've talked about our MHC class 2, because B-cells can also be antigen-presenting cells, which bind to T-cell receptors on CD4 T-cells. We have CD40, which we'll talk about later, but this interacts with CD40L on the T-cell to induce antibody class switch. Then we also have B7 on our B-cells, which was CD8 and CD86 for B7-1 and B7-2. And these are costimulatory molecules used when we interact with our T cells. So if you guys remember, these are important T cell function. And then we also have our CDLA, which can bind to this to inactivate T cells. And then we have CD32, which is low affinity Fc receptor for immunoglobulin G. So again, these are the key molecules expressed at the surface of the mature B cell. Again, activated B cells express more class 2 MHC and B7, so we can have better interactions with T cells. So the B receptor complex is our basic BCR in our Ig alpha, Ig beta. Then we have our co-receptor, which is CD19, CD21, CD81. Then we also have CD40, which interacts with CD40 ligand on T cells, which causes some class switching. Then we have our CD32, aka Fc gamma receptor 2b. This is the low affinity Fc receptor for IgG. Then we also have a negative, which has a negative role for downregulating B cell activation. So B cells can also present antigens to T cells, as I've just said. So antigen can also be internalized by the B cell receptor complex. This antigen is then processed and presented by class 2 MHC. B cells can act as professional ABCs to stimulate T cells. So the B cells can use surface immunoglobulin to present specific antigen very efficiently to T cells. So surface immunoglobulin allows B cells to bind and internalize specific antigen very efficiently, especially if the antigen is present as a soluble protein. The internalized antigen is processed in intracellular vesicles where it binds to MHC class 2 proteins and is transported to the B cell surface where the peptide MHC class 2 complexes can be recognized by T cells. So this is kind of going back to what we've already talked about with T cells interacting with B cells. Now we're kind of going the other way. So when the protein antigen is not specific for the B cell receptor, its internalization is inefficient and only a few protein fragments are then presented at the B cell surface. So again, antigen-specific B cell binds. Specific antigen efficiently internalized by receptor-mediated endocytosis. It's being endocytosed, then it's going to match our MHC class 2. Then this high-density specific antigen is presented on our MHC 2. So B cell activation usually requires a series of signals. So this is kind of a summary. So there's signal 1, which is delivered when the antigen binds to the immunoglobulin/B cell receptor in the B cell membrane. So here's that step 1. This signal is boosted from the B cell co-receptors, not shown here, but you know, the CD19, CD21, and the other ones we saw. And we also have additional signals, which are also delivered by T helper cells. So our T helper cells is binding to essentially, this is the same antigen, and it's binding with our antigen-presenting cells. So this is a macrophage or dendritic cell, and this is also stimulating these B cells through signal 2. These are usually nearby, responding to the same antigen. So B and T cells can recognize the same antigen, but in different ways. They usually see different epitopes of an antigen. So our T cells see peptide fragments, complex with MHC proteins, often internal epitopes, and our B cells see free antigen, often surface epitopes. So here we see an APC is activating a T helper cell, which in turn activates a B cell. The B cell then differentiates into antibody-producing plasma cell. So, as we hear, so this was a sample of a T cell independent. So this T cell was activating this B cell. So now we're going to look at T independent antigens, also known as TI. So there are two types of T independent antigens, T1, TI1, and TI2. So what are the characteristics of these B cells that are T independent? So they mainly have IgM and have little or no class switching, which again is a main function of T cells and their cytokines. And there's also poor induction of memory cells. So they're kind of a type of adaptive immunity, but with innate function. So they're used in they're secreted very fast, but again, they don't have memory. So they kind of mainly work in neutralization and complement activation, mainly due to the fact that it releases IgM. So these thymus-independent 1 antigens activate B lymphocytes binding to TLRs rather than B cell receptors. So essentially, it's an antibody response in an innate way, but it's still through antibodies. So again, here's an example of TLR4 binding to LPS, and we also have our B cell receptor, or a TI1. Our TI2 antigens activate B lymphocytes by simultaneously cross-linking many B cell receptors. So here's a long polysaccharide and repeated subunit polysaccharides. So here we can see a bunch of cross-linking occurring. So now we're looking at thymus-independent antigens again. So again, our type 1 antigens can recognize LPS and bacterial nucleic acids. These can activate B lymphocytes by binding to receptors other than the B cell receptor immunoglobulin. So examples are TLRs, like we've talked about, such as LPS binding to TLR4, which can cause the transcription of antibody genes. T-cell independent antigens, such as LPS, activate B cell specific for LPS and B cell specific for other antigens. The signals delivered by these LPS through the TLR4 binding activate B cells, and T-cell help is not needed. So again, T-cell independent. So LPS is a T1 antigen that activates LPS. LPS-specific B cells, as we can see here. Again, activation, proliferation, differentiation. IgM antibodies specific for LPS are released. LPS also activates B cells specific for other bacterial surface antigens in a T-independent manner. So again, there's further activation. Then IgM antibodies specific for other components for the bacterial surface are also released. So here's a little antibodies being released. Then bacterial DNA. Bacterial DNA is also a T1 antigen that can help activate B cell specific bacterial surface antigen. And again, we have our activation, proliferation, differentiation. Then more IgM antibodies specific for bacterial surface antigen. So our TI2. So our TI2 antigens have multiple repeating subunits. So example would be capsular polysaccharides. So here's an example. These capsular polysaccharides activate B cells by simultaneously cross-linking many B cell receptors. So here you can see many cross-linking, and this cross-linking activates the transcription of antibody genes with little or no class switching and no B cell memory, and is mainly induced at IgM production. The conclusion is essentially that T cells must be important in generating memory B cells and in helping B cells class switch. So if you only have, we can have, like we've talked about, you can have defects in this class switching, which results in people mainly producing IgM, but they still end up being very susceptible to certain things like viruses. So type 2 thymus-independent antigens engage many B cell receptors on the same specificity on a B cell surface, cross-linking the membrane immunoglobulin and activating the B cell, so it does not depend on additional signals from the T helper cells. So interactions between B cells and T cells are needed for full B cell activity. So essentially, here's a summary figure of our B cell and T cell activation in interaction. So first, we'll have our B cell activation to the B cell receptor. So again, here we have our signal, and here's an example of T cell TLR working. Then we have our activated B cell, which increases expression of B7. So again, this is needed to communicate with the T cell, which needs its secondary signal. Then they'll have T cell activation through T cell receptor and co-stimulation via CD28 with the interaction between B7-1 and B7-2. And then this T cell will make IL-2, which stimulates T cells further. So remember, IL-2 works on itself. And then, yeah. So this is a walk through the steps. So now we're going to move to the next step. So again, we got to 3, where it ended up in IL-2 being released to stimulate T cells further. So next, this activated T cell increases CD40 ligand expression. So again, here's our CD40. Then we have CD40 ligand, which binds to CD40 on the B cell, which sends signals for class switching, which boosts the B cell and T cell interactions further. Then later, we have T cell, which increases the CDLA4. And again, this is kind of to shut it off after it's been working hard, doing its thing. So the CDLA4 interactions with CD80, which has CDLA4 has higher affinity for that receptor. And this downregulates the T cell activation, bringing the immune response to it, bringing it to an end. So these are, again, needed for full B cell activity. So we have antigen recognition induces CD40 ligand and cytokines by the Th2 cells, for example. This can be other T cells as well, which activate the B cell. B cell proliferation, differentiation, to antibody-secreting plasma cells. So again, and this will be isotype switched in most cases. And in Th2, this is likely IgA or IgE. So T cell help is delivered through the CD40L, binding through CD40 on the B cells through cytokines produced by T cells, results in proliferation and differentiation of B cells. And that's the end, guys. So that one's a little confusing in spots, but I recommend just slowing it down. And again, really follow the figures, because those are like the meat and bones of the lectures, because I'm essentially just talking about them in a longer version in the text. So yeah, definitely slow down and take a look at the figures. So again, even here, this can summarize most of the interactions we talked about. But yeah, I hope you enjoyed it. And then I think next we're going to look at the different types of T cells. But yeah, see you next time.